Dear student, today we will discuss about the probes in molecular biology. So what is probe? Okay. So probes are the known short single-stranded labeled sequence of RNA or DNA used to detect the presence or absence of its complementary sequence in biological sample. Now, each and every word is important here. Let's understand. Probes are known. Known means we know the sequence of probe because it is artificially synthesized and this is tagged with some labeling. Okay, this is how the probe is synthesized. We will see how it is known. And short means about 10 to 10,000 base pair. Single stranded, why it has to be single stranded, as the detection of the probe is based on in vitro hybridization. Therefore the probe prepared is always a single stranded. Otherwise, if it is double stranded, then no hybridization will occur. And then it has to be labeled. Labeled means labeling is done for the detection of target sequence. This may be isotropic, non-isotropic, or fluorescently done. If there is no labeling done, then detection of hybridization is not possible and no probing can be carried out. Okay, then sequence of DNA called DNA. If it is DNA sequence, we say DNA probe. If it is RNA sequence, we say riboprobe, used to detect the presence or absence of its complementary sequence. Now, what is complementary sequence? Complementary sequence means the sequence of the target or specific DNA that we are looking for in the sample under the testing. So we have a sample and we are looking for some target DNA sequence. We don't know whether it is there in our sample or not, so we will use the probe. Okay, and these probe sequence will hybridize with the target sequence because they are complementary to it. So complementary sequences are there. So again, if I wrote, probes are the known short single stranded labeled sequence of RNA or DNA used to detect presence or absence of complementary sequence in biological sample. Okay, now we will see the labeling first, the labeling part of the probe. So there are radio labeled probe, non radio labeled fluorescent, and others also. We'll discuss just three. Radio labeled probe, the probe are labeled with the radioactive molecules such as P32, S35, H3, I125, and there are ways to label like to do the labeling like nick translation, or either they are done at the end. Um, but these are the main you can say radio labeled probe which are used, and they are detected by autoradiography. They are has high sensitivity, that is, if the probe is used at very low concentration, the target hybrid can be easily detected. So they have high sensitivity, the radio labeled probe, but there is high risk in using since they emit radiation, so safety measures are high. If you look at the non radio labeled probe, these probes are labeled with enzymes such as alkaline phosphatase or horse radix peroxidase. The enzyme is attached to the probe and its presence is detected by use of substrate that changes the color. We have seen this in ELISA. The enzyme is also known as reporter probe. Okay, so here the probe is labeled in some other way, in the indirect way. That probe is attached with the enzyme, whereas in the radio labeling, this is directly incorporated in the probe itself. Okay, now fluorescent probe is again the probe is attached with some fluorescent chemical in this. The fluorescent is detected which is directly proportional to the hybridization. So fluorescence is detected like that. So fluorescence in situ hybridization, that is FISH, is done to examine microbiological or cytological specimen under microscope. Some of the fluorescent probes are Tagman probe, Scorpion probe, molecular beacon, which are commonly used as DNA probes in real-time PCR. We'll just discuss the Tagman probe. So it has 5 dash 3 dash sides. At the 5 dash side, fluoro 4 is attached, which is a fluorescent dye, you can say. And at 3 dash, quencher is there, quencher molecule is there. So at 3 dash and it has quencher, and 5 dash and fluorophosphor, which is known as reporter, is attached, and they are in close proximity. Once the DNA probe is attached to its target sequence, it bind. Okay, and at the same time, the 5 dash exonuclease activity of DNA polymerase will cleave the reporter, and after detaching, it emits the fluorescence. So what is happening from the 5 dash end? This reporter is detached by the DNA polymerase. DNA polymerase also has the exonuclease activity, and once this is detached, then it is going to emit the fluorescence. This fluorescence is detected by a detector. Okay, and this determine what? This determine this fluorescence activity is directly proportional to the hybridization of probe with target DNA. Okay, so this is about the labeling of probe, and we will see now the hybridization with the probe. So there are different techniques that are used for the hybridization of probe: southern blotting, northern blotting, colony hybridization, plaque hybridization, fluorescent in situ hybridization, sequencing by hybridization. Now, in the southern blotting, it is used to detect the specific DNA. So, which hybridization is there? Here, the DNA, target DNA is hybridized with the DNA probe. Okay, this is called southern hybridization. If the RNA is hybridized with DNA probe, we say it is northern blot. Okay, and in colony hybridization, the bacterial colony or gene libraries in plasmids are screened to search gene of interest. In plaque hybridization, the viral colony or phage libraries are screened to search for the gene of interest. So, there are randomly selected many, you can say, um, copies of genes, and they are all hybridized with a single probe. So wherever the hybridization occur, that is the gene of interest. And which we have already discussed in real time PCR. I'll be discussing sequencing by hybridization. So it is cloned by using the random primer probes to identify certain sequences that occurs repeatedly. So there are certain repeatedly sequences like VNTR, and they are used as DNA marker to identify the nearby genes in the genome. So usually what done, these sequence tag sites are there, for example. Okay, so we can use the primer, which is used as probe, and this probe actually hybridizes certain part, and then we can carry out the we can sequence, or we can carry out the hybridization plus uh, the polymerase chain reaction, and it is going to copy some part of that repeated sequences. Okay, and once we come to know that a particular cell has these sequences, then we can also find out that which all genes are occurring nearby these repeated regions. So thereby it helps to do physical mapping and genetic mapping in the genome. So this is all about hybridization, and we'll just see the methodology how it is done. So here you can see either from the DNA library or from the cell we take out the DNA and then it is treated with restriction endonuclease. We obtain DNA fragments. They are run on the gel, then transferred on the nitrocellulose filter paper, and then hybridization is done. So DNA is on the filter, nitrocellulose filter, whereas um, label probe is in the liquid medium. And wherever you see the hybridization, okay, this is detected by autoradiography. So this is how the probes help in the detection of a specific part of DNA or target DNA. Now, what are the application used in the medicine? Detection of genetic disorder and detection of tandem repeats. In the medicine, probes are used to detect presence of growing virus in human body, as viruses are not easily cultured on artificial medium. We have already seen in real-time PCR how the probe is used. Then detection, here fluorescent probe is used. Now in the detection of genetic disorder, the diagnostic probe against phenylketonuria, cystic fibrosis, sickle cell anemia are used to identify changes in the sequence of original or normal gene. So if the normal gene changes or get mutated and have some different sequence, then that could be easily detected by using the probe. Now, detection of tandem repeat. So here the VNTR and other tandem repeats are detected by using primer probes in combination of DNA fingerprinting, and they are also used in the forensic science and paternity test. So here also we use the probes. So they have wide application. So this is all about probes in molecular biology.